So starting with the divisions of the nervous system, we have the central nervous system, which is really the brain and the spinal cord, so the central structures. Um, and then we have peripheral. So peripheral is everything else. It's going to be things like nerves, receptors, ganglia, right? So all things that are outside of the brain and spinal cord. When we think about the structures that protect and house the brain, these are called meninges, and these are on the outer side of the brain. There are three parts to the meninges, actually. There's the dura. The dura is the thicker, tougher outer layer. It's actually connected to the skull, right? If you look at the skull bone here, the inner connective tissue layer that you're seeing here is called the dura. Now, the dura is going to have modifications or inflections that create these structures called the falx cerebri along the center of the brain. So the falx cerebri is really a division or a fold of the dura, the dural fold or dural reflection. And so it goes down in between the cerebral hemispheres and separates the right and left hemispheres. So we call this structure here the falx cerebri, right? Separating the cerebri. Now, the tentorium cerebelli is a similar reflection of the dura. It actually goes up and separates the occipital lobe, which is part of the cerebral hemisphere on the top, from the cerebellum, excuse me, which is the lower part of the brain itself. It's a separate part of the brain. So the cerebellum is underneath, and then the occipital lobe is going to be on top, right? And that is going to come inward and separate both. So these are two dural reflections that help to separate some of the brain regions that we're going to talk about coming up. Now, inside of the dura, the deeper layer is called the arachnoid mater. And the arachnoid mater is really not as opaque or as thick as the dura. It's much more of a network or sort of a meshy layer where we have a lot of capillaries and arteries and veins, okay? And so that arachnoid mater is going to then separate the inner layer, which is called the pia mater, from the spinal cord. So in between the pia mater and the arachnoid mater, we're going to call that the subarachnoid space. And that is a really important layer where we have cerebrospinal fluid, which helps to protect the brain and spinal cord. So the dura mater is the outer layer, arachnoid mater is the middle layer where we have capillaries and venules. And then the pia mater is the innermost layer, which is really one with the brain itself, connected to the brain. Let's look at the cerebral lobes themselves. So again, looking at the brain, and if you ever were to hold a brain, it's really quite mushy. And so it really does need those meningeal layers to protect it from the rough features inside the skull, right? From moving around and sort of being damaged against the walls of the skull. It's actually when we harden it in the cadaver lab with like formalin that it actually becomes a little bit more holdable without actually kind of coming apart in your hands, right? It's really soft and delicate. Now, the cerebral lobes are going to be divided functionally. So there are different lobes that control different regions of the body, as well as different functional aspects of our personality, things like memory, things like emotion, right? Different aspects of the way that we are, right? Our personality. And so if you look at the names of the lobes, we'll talk about each of their functions as well, but they're going to be divided by some of these grooves in the brain that are called sulci to create these bumps that are called gyri, right? So this depression itself is called a sulcus, whereas the bump that's created from the depression is called a gyrus. And so there are sulci that separate lobes and fissures that are larger sulci, as well as the bumps that are created that are functional regions of the brain itself. All right. The parts or the lobes are also going to be named according to the bone that they come in contact with. So the frontal lobe is in the frontal bone. Parietal lobe, which controls things like speech, will be against your parietal bone of the skull. Temporal lobe is going to be deep to your temporal bone of the skull. Temporal lobe is going to be associated with things like hearing, as well as memory and emotion. Occipital lobe is going to be the posterior-most part. This is really important for vision, and that is going to be deep to the posterior bone of the skull as well. Okay, so each lobe is sort of connected to the appropriate bone. However, bear in mind that for the frontal lobe, for example, there's only one frontal bone. But for the frontal lobes of the brain, we do still have two frontal hemispheres, right? Two frontal lobes. Same thing for the occipital lobe. There's only one occipital bone of the skull, but we do have a left and right occipital hemisphere. Let's look at some of the sulci, gyri, and bigger or deeper sulci, which we call fissures. So gyri are the ridges that are created from these depressions or grooves. Sulci are the grooves themselves, right, that create these depressions. And then fissures are the really deep grooves like the longitudinal fissure. So the longitudinal fissure is where we find the falx cerebri. So we talk about the falx cerebri being that the dural fold that goes down in between the cerebral hemispheres, but the depression in which it sits is called the longitudinal fissure, right? And it's really deep. We also want to think about the right and left hemispheres. So according to this division, we have the left hemisphere of the brain and a right hemisphere of the brain. Now, usually we're going to have a dominant side of the brain, right? The left hemisphere is usually the dominant side, which makes most people right-handed, right? You have a little bit more dominance, a little bit more strength in your right hand. But we do have left-hand individuals, which means that they have a more dominant right hemisphere. Okay, and then different sides of the brain will have different structures. And so it's really important to think about dominance when we think about things like strokes and so forth. That becomes really important. Now, the cerebellum is going to be another separate part of the brain, separate from the cerebral hemispheres themselves. So all the lobes we just described together form the cerebrum or the cerebral hemispheres. And then the cerebellum is going to be underneath the cerebrum, right? All of this is cerebrum, and then this is cerebellum. The cerebellum is literally translating to tiny cerebrum. So it's really a smaller version, mini-sized version of the cerebrum, which is much larger. And then it's going to be underneath the occipital lobes and sitting in front of the brainstem structures, which we'll talk about coming up. Now, the function of the cerebellum is really involved in things like our coordination, our balance. They're going to control our motor activities. So this is what helps you to have smooth coordination. And so this is why alcohol, which particularly affects the cerebellum, makes it very difficult to coordinate your movements to maintain your balance, right? It's going to throw your equilibrium off because it affects your cerebellum. And that creates a gait disturbance called ataxia, which is where you can't walk in a steady gait. So that is kind of the functional connection with the cerebellum. Now, the brain stem is going to be the central portion that leads down to the spinal cord. It's going to be in direct connection to the cerebrum, which is the outer portion that we just talked about, where we have the lobes. And so it's going to be sort of a central highway of communication between the cerebrum on top, the cerebellum, and the spinal cord, which controls the limbs for things like movement, right? So the brain stem is going to be the primitive or sort of early lower part of the brain, less evolved part of the brain. It's connected to the diencephalon, which is right above the spinal cord. So the diencephalon is really kind of where we have the thalamus, central parts of the brain. And then that leads into the midbrain, also called the mesencephalon. That is followed by the larger sort of swelling called the pons. And then that is a continuation with the medulla oblongata. And the medulla is continued as the spinal cord. So this is the three central regions together that form the brain stem. Okay. Functionally, they're going to have nuclei, which are clusters of cell bodies that create connections to our face and our limbs for movement, for coordination, for a lot of nervous system function that allows us to move and, coordinate within the world. So really important for coordination through some of the nuclei that we find here. Let's talk about the individual parts here. So the midbrain or mesencephalon is the superiormost part of the brainstem. It is going to help control regulating our sleep patterns, regulating our vision and hearing, and then things like our reflexes, right? This is why we make the appropriate audio and visual responses as a part of our reflex. So if someone taps you on your shoulder, if you hear a sound on the left or right side of the room, your reflex is to move your vision to that part of the room. Same thing with your hearing. You're going to attune your hearing to that part of the room. So that is some of the function that is associated with the midbrain and the mesencephalon. The pons or medulla. So the pons are going to be the second part of the brainstem, right? So this more larger region. And this is where we have the largest part of the brainstem. This is where we have structures that are also involved in our audio and visual regulatory responses. And this is going to be continuous with the medulla and then eventually the spinal cord. So pons will be the larger portion, medulla would be the smaller narrowing, and then the spinal cord. Okay, let's talk about the circle of Willis, right? I'm going to take a brief pause here to see. Okay. Now, the interesting part of the circle of Willis is that it really creates this circle which is supposed to maintain continuous flow and keep the brain perfused even if there's a blockage on any side, right? So it's what we call an anastomosis. An anastomosis is an area where the arteries create alternate routes of flow, which are kind of like a backup reservoir, a backup source just in case there's a blockage. So we want to think about the posterior aspects of the circle, and then we'll talk about some of the anterior branches. So posteriorly, we have the vertebral arteries. And the vertebral arteries are branches of the subclavian artery. We talked about the transverse foramen of the cervical vertebra. This is where the vertebral arteries will traverse. Then they're going to run on top of the brainstem, on top of the medulla. Then they're going to merge to form the basilar artery. So together, they're going to form the central basilar artery, which is located anterior to the pons, right? We just talked about the pons. Now they're going to branch off at the junction between the pons and the midbrain. They're going to branch off to form the posterior cerebrals, which basically perfuse the occipital part of the brain, right? The occipital lobes. The posterior cerebrals are then going to be connected to the internal carotid via the posterior communicating, okay? So there are these two very tiny communicating branches that are going to connect the posterior cerebral to the middle cerebral and the internal carotid. And so this is what starts to create this circle-like structure that allows for flow to move in different directions just in case there's a blockage. So now at the central part, we have the internal carotid. Now the internal carotids are coming up from the anterior aspect of the head and skull, right? They're coming up by way of the arch of aorta, then they come off as the common carotid, and then they branch off external, supplying the face and skull, internal coming to supply the brain, coming up to supply the brain. So the internal carotid is going to be an anterior route of supply, whereas the vertebrals are going to be a posterior route of supply. So just in case there's a blockage on either side, there can still be perfusion to the brain, right? So the internal carotid, and we're only seeing the cut surface of this because we're looking at the brain sort of inverted here. We're seeing a cut surface of the internal carotid, and then we're immediately seeing the branch to create the middle cerebral, right? So here's the middle cerebral, which goes and supplies the temporal lobe and a part of the frontal lobe as well. And then from there, we have the anterior cerebral, right? So here's middle cerebral, here's anterior cerebral. The anterior cerebrals are going to go towards the front of the brain, and they are going to supply the frontal lobes, but they're also going to be connected across the midline by the anterior communicating artery. right? So whereas we saw two posterior communicating arteries, one on both sides, anteriorly, we only have one anterior communicating artery that connects both the right and left anterior cerebral artery, right? And then the anterior cerebral arteries will go forth, go towards the front region to supply the frontal lobe of the brain, right? And so you can see how that creates this circle, right? So just in case it's a blockage to the carotids, the vertebral arteries will keep most of the regions perfused. And if there's a blockage to the vertebral, the carotids can keep the brain perfused. Now let's talk about the sinuses. And the sinuses are sort of the venous alternate, right? These are the venous structures within the brain. These actually create these larger channels than we find in other areas of the brain. This is why we call them sinuses. They're much larger and they're sort of embedded within the layers of the dura mater, right? That thicker outer layer of the meninges. Now, the other unique feature of the sinuses is that they don't have valves, so they have much more continuous or smooth flow. And then they are basically embedded within different parts of the skull internally as well. Now, they're uniquely named in different regions. We're not going to look at the different names, but they do sort of drain in a unique pattern that brings all of the venous drainage of the brain back towards the internal jugular vein that then goes back towards the heart. So this is how the venous drainage of the brain can be returned to the general circulation and then go to the heart. If we look at some of the specific venous sinuses here, we have the superior sagittal sinus. Now, the superior sagittal sinus is running within the falx cerebri here, within the longitudinal fissure, that groove within the cerebral hemispheres. We have the confluence of sinuses. Again, this is where most of the posterior sinuses will meet. Again, we're kind of having a drainage pattern, kind of like a collection of rivers, if you will, that is bringing it to a central point. So the confluence is a meeting point where we have the superior sagittal sinus on the top meeting with the sigmoid sinus, and then eventually connecting the transverse sinus as well as the deeper occipital sinus, okay? So all of these sinuses come together at the confluence of sinuses. And then again, the drainage is going to come to the confluence and then